Good morning and good afternoon, everybody, and welcome to this first joint e-seminar organized by the Japanese Society of Nephrology and YNP, or Young Nephrologist Platform of ERA-EDTA. My name is Martin de Borst and I am the chair of YNP. YNP is a platform which aims to involve young European nephrologists in all the activities of European nephrology and of ERA-EDTA in particular. YNP was established in 2013, and it has been our dream to connect young nephrologists and nephrology researchers within Europe and to connect with young colleagues from all over the world. Today, we are taking a new step with this e-seminar that we organize in collaboration with our Japanese friends from the JSN. And I am honored to open this meeting that will feature 4 excellent clinical and basic scientists. A special welcome to the presidents of ERA-EDTA, Professor Wanner, and the president of the JSN, Professor Kashihara. We are thrilled to have you in our meeting on this special occasion. So now, before we are going to start, let me inform you that in case you have any questions during the meeting, you can ask these using the Q&A button at the bottom of your screen. I will now hand over to Dr. Motoko Yanagita, who is the chair of the Committee of International Affairs at JSN. Dr. Yanagita.
Thank you, Dr. de Borst. And thank you very much for giving me the opportunity to address at the beginning of this meeting. First of all, congratulations on the successful organization of this first joint meeting. JSN and ERA-EDTA signed a collaboration agreement in 2015, and since then, we have been exchanging speakers in our congresses and collaborating in basic and clinical research. Ambassadors from each society have increased the visibility of their own society and provided opportunity for future collaboration. However, until now, the exchange have been limited to prominent professors. In order to further strengthen the ties and collaboration between the two societies in the future, it is important to have an interaction between various generations. For this purpose, President Wanner and President Kashihara kindly set up this new platform. This first joint meeting was planned and was organized by young nephrologists in YNP and its counterpart in JSN. We hope that this meeting will strengthen the collaboration among the young generation and become a basis for future collaboration. Finally, I'd like to thank Monica, Isabel, and Pepe for their efficient and kind cooperation as always, and ERA-EDTA for providing the platform for Zoom. Thank you very much, and let's enjoy the meeting.
Thank you very much, Dr. Yanagita. It's now time to hand over the word to the president of the ERA-EDTA, Professor Wanner. Welcome and thank you for joining us.
Thank you. First of all, again, thank you for giving me the opportunity to address some words to this new platform on behalf of the ERA. And I want to thank, first of all, President Naoki Kashihara for his strong support for this new initiative. And I think this has been built on friendship and trust, which has been built up over the last years, where we had, as Professor Motoko Yanagita told us, had some lectures in Japan and at the ERA from prominent professors. But this new program was developed also to give some visibility to the young scientists and to understand the two programs in Japan and also to learn from each other. Giving visibility to young nephrologists is, I think, very important. And therefore, I thank you for Professor Motoko Yanagita and Martin de Borst for having organized this meeting. And thank you also to the chairs, Professor Orsolya Cseprekál and Shinya Nakatani, which will chair this meeting today. So thank you all, and I wish this meeting a good success and hand over to President Naoki Kashihara, please. You are on mute, Naoki.
Sorry. Thank you very much, Professor Christoph Wanner. My name is Naoki Kashihara, currently serving as the president of Japanese of Nephrology. So first of all, I'd like to express my sincerest gratitude to Professor Christoph Wanner, ERA-EDTA president, and Dr. Martin Borst, YNP chair, and all the faculty members of ERA-EDTA. In JSN side, Professor Motoko Yanagita, Chair of Committee of International Affairs of JSN, and Professor Masaomi Nangaku, Executive Supervisor of JSN, and also the President of APSN, Shinji Kume and Shinya Nakatani. These members have worked hard in many days to prepare this meeting. The relationship between ERA-EDTA and JSN has grown closer in recent years, thanks to Professor Wanner's strong leadership and support. We are very grateful for this. I believe to make this bilateral relationship closer and stronger, the relationship needed to become more multi-layered. So in this sense, we are very pleased to be able to organize such a seminar where young nephrologists from both societies can meet and exchange their ideas and communicate directly. I believe the future of our societies depend upon these young nephrologists. So I hope this seminar will continue for many years to come. Thank you very much.
Thank you very much, Professor Wanner and Professor Kashihara. We are honored as young nephrologists to have the support of the presidents of our both societies. I think this is a very important signal. And so thank you very much again for your attendance and for your kind words. Now it's time to move to the presentations, the 4 talks, 2 basic talks and 2 clinical talks. And for that, I will now hand over to the 2 chairs, Dr. Nakatani from Japan and Dr. Cseprekál from Hungary, from Europe, to chair the remainder of this e-seminar. Thank you.
Thank you very much, Martin. It is my pleasure to also welcome everyone, all the attendees and all the panelists. So we have arrived at the main part of our first joint e-seminar. We are going to have altogether 4 presentations. 2 presenters are coming from ERA-EDTA, and 2 presenters was proposed by the Japanese Society of Nephrology. One senior and one excellent young researcher will going to present us their work. So I would like to encourage all the attendees to ask their questions in the chat boxes and to the presenters immediately after the presentation. So I also would like to welcome my co-chair, Dr. Nakatani, and I'm giving the word for him to present, to call our first presenter today. Thank you.
Orsolya, thank you very much for your introduction. Hi, good morning, good evening, everyone. I'm Shinya Nakatani from Osaka State University. So it's my pleasure. I'm acting as co-chairperson of the presentation session. So it's a pleasure for me to introduce Professor from Erasmus Medical Center, Rotterdam, the Netherlands. As you know, his specialty is on the mechanisms of renal tubular transport and the link with human disease, include electrolyte disorders, hypertension, and CKD. The title of this presentation today is Urinary Extracellular Vesicles, Small but Mighty. Could you start your presentation, please?
Thank you very much for the kind introduction and the invitation to speak in this joint session. It's an honor and a pleasure. And what I would like to discuss in this first presentation is a very exciting field, I think, the field of extracellular vesicles. That is exciting for biology and medicine in general, I think, but also especially for nephrology when it comes to urinary extracellular vesicles. And I'm happy to give an introduction and a brief overview of our activities in this exciting field during this presentation. I have nothing to disclose, and I would like to start with this painting by a Dutch painter who was actually a student of Rembrandt van Rijn, with whom you might be more familiar. But I think it very elegantly illustrates the old tradition of uroscopy, trying to establish a diagnosis by analysis of the urine, which is of course one of our daily activities. And there has been evolution in our diagnostic approach using urine, of course, although perhaps not as molecularly based as we would like it to be. It's still mainly urinary albumin and some other markers. And I think especially the field of urinary extracellular vesicles holds great promise to introduce more molecularly based and disease-associated biomarkers into nephrology. So the outline of my presentation will be as follows. I think it will help to give a brief overview. What are these extracellular vesicles? Why would we have an interest in these vesicles, and how can we use them? The second part will be studying the UEVs as a source of biomarkers in kidney disease. And finally, which is perhaps the most intriguing, but also the most challenging field and least evolved, I would say, UEVs as mediators of kidney function and perhaps also contributing to certain kidney diseases. So starting with the first part. So urinary extracellular vesicles is currently the official nomenclature for all vesicle species and populations present in urine. These are typically nano-sized vesicles, ranging from approximately 1,000 nanometers to less than 100 nanometers. And the three main populations of vesicles that can be identified in urine are exosomes, which is a term you might be familiar with, and is perhaps also the most interesting subpopulation, I think, because they result from the fusion of multivesicular bodies, and after fusion of these multivesicular bodies with the plasma membrane, cells release these exosomes in urine or other biofluids, and they really give a capture of the cytoplasm and all of the proteins that are in the cytoplasm. Another category is microvesicles, which bud directly from the plasma membrane. And finally, there are also apoptotic bodies. They differ in their characteristics. For example, microvesicles are larger, approximately 1,000 nanometers, whereas exosomes are on average 150 nanometers. But all of these subpopulations of vesicles contain both proteins, but also DNA, RNA, and various metabolites. So there is a particular interest in exosomes, as I alluded to, and there are certain hallmarks of exosomes that are important also for the remainder of this presentation. There are a number of putative surface markers for exosomes, certain tetraspanins shown here. Most notably CD9 and CD63 that can also be used to isolate exosomes. And they contain a number of exosome-specific proteins that are related to their exocytosis process. Heat shock proteins, TSG101 and ELIX are examples. And you also see illustrated here the content of these exosomes that is, of course, of interest when searching for novel biomarkers. Transmembrane proteins are also present in exosomes, and it's important to emphasize here that they have the same configuration as in kidney epithelial cells. There are also vesicles and exosomes with reverse topology, but the majority has the same orientation as in the kidney epithelial cells. So this slide intends to illustrate why it might be important to study urinary extracellular vesicles. This is from a previous study that we conducted a few years ago, where we performed mass spectrometry on either whole urine, identifying over 1,000 proteins, and using mass spectrometry to analyze the proteome of urinary extracellular vesicles, also identifying a bit over 1,000 proteins. And as you can see in this Venn diagram, there is a certain overlap in these proteins, but there are also proteins that are only identified in urinary extracellular vesicles. And many of these proteins are very relevant, I think, to nephrology because they are disease associated. And as alluded to before, I think they provide a more molecularly based analysis of kidney physiological or pathophysiological processes. The methods, especially the mass spectrometry methods, are ever evolving. And so currently it is possible to identify over 2,000 or 3,000 proteins in the UEV proteome. So that's the speed at which these technologies are improving. So there are several methods, and it's important to take this into consideration because the method that you apply when isolating or characterizing vesicles also determines very much so your output. Most people in our lab does so as well use differential ultracentrifugation, which is a rather robust method to isolate and precipitate vesicles. But other methods are available, including ultrafiltration, precipitation, and affinity isolation, including immuno-isolation methods. It has become increasingly important also to quantify the number of vesicles. The most commonly used method is nanoparticle tracking analysis, using a nanosite as a device to do so. But there are also other methods that have been recently developed. This is one we developed here in-house with a colleague from pathology and urology called EV-Quant. And of course, and this is an issue in nephrology in general, especially for higher throughput, it would be elegant to use spot urines and not 24-hour urines. And then the issue of normalization becomes important. So if you do not want to have a timed urine collection, you need a method for normalization. This is more or less the holy grail in the UEV field. And people have used various approaches, including exosomal markers like ALEX or TSG101, but perhaps urinary creatinine, similar to albuminuria and the albumin to creatinine ratio, might offer a much more efficient approach. But until recently, it was not clear if this could be used to normalize the UEVs. And then finally, for characterization, you have a myriad of methodologies. Electron microscopy is important. Most studies used it just simply as a visual confirmation that vesicles are present in the biofluid or urine. But other methods with higher throughput possibilities include flow cytometry, mass spectrometry, immunoblotting, and mRNA. And obviously, you would like to go to a more immunoassay or ELISA methodology if you really want to achieve high throughput towards clinical application. And as alluded to at the beginning of this slide, methods differ in the yield of the number of vesicles. The detection limit differs, the throughput and the type of EV subpopulations that you isolate depends on the methodology used. So this is a recent paper we published just a few weeks ago addressing this issue of normalization, quantification, and characterization of urinary extracellular vesicles. And just to give you the main conclusions, We did find that UEVs in spot urine can be reliably normalized by urine creatinine, opening up a window for higher throughput analysis of UEVs. We also identified a number of caveats when using certain techniques. So we found, for example, that in more dilute urine, there is a bit of swelling of these vesicles because they indeed contain all of the transport proteins, including water channels. And this might affect how UEVs are quantified. We also identified interference with uromodulin, and this might then interfere with the quantification of vesicles, especially when using nanoparticle tracking analysis. And until recently, this was the method that was recommended also by the International Society of Extracellular Vesicles. But increasingly, it becomes apparent that this method has certain limitations that need to be taken into account when quantifying vesicles. Also important, especially towards developing immunoassays for higher throughput applications, we found that it is possible to use detergent to gain access to the vesicles, because most antibodies as we use them are intracellular epitope antibodies. And because of the same configuration as in kidney epithelial cells, you do need to gain access into the vesicle to be able to use these intracellular epitope antibodies. And this can be done reliably with a low concentration of detergent without interfering with the stability of the UEVs. And finally, we also found that, and we did so by analyzing the localization of CD9 and CD63 along the nephron, that you can use these markers to capture nephron segment-specific vesicles, which might be relevant if you would like to study disease in a nephron-specific segment. So this is illustrating an approach that we also developed here in-house, where we use a biotinylated anti-CD9 antibody to capture and isolate these vesicles. As mentioned, if you want to use intracellular epitope antibodies against your protein of interest, you need detergent to gain access to these vesicles. And then you can introduce a second antibody against your protein of interest that you can illuminate using fluorescence, mostly, for example, using horseradish peroxidase. And another possibility is to use a third antibody to also at the same time quantify the number of CD9 positive vesicles using a different label, for example, europium labeled third antibody. So this is an approach that we developed to develop an immunoassay for higher throughput application and analysis of these UEVs using a targeted approach against a protein of interest. So then moving on to the second part of the presentation, virtually all kidney diseases have been studied now for UEVs, and I just would like to highlight a few of them. I think it's very important, which is another holy grail, if you will, to see if these UEVs recapitulate normal renal physiology, meaning that what happens in the kidney is also directly reflected in UEVs. And we studied this by analyzing the relationship, the emerging relationship between plasma potassium and its effect on a certain sodium transport protein in the kidney that we have an interest in, the sodium chloride co-transporter, the thiazide-sensitive co-transporter, and especially its phosphorylated or active form. And in kidney tissue, this has now been very nicely established that there is a direct correlation between the plasma potassium level and the abundance of phosphorylated NCC in kidney tissue. And then as confirmation, several studies have used and isolated UEVs, used immunoblotting to analyze phospho-NCC in patients, for example, patients with primary aldosteronism, so high aldosterone levels, aldosterone and potassium being regulators of phospho-NCC, again establishing this linear relationship similar to the relationship that was identified in kidney, illustrating that indeed, at least for this protein, there is a one-to-one relationship between what occurs in the kidney and what is being reflected in these UEVs. We also confirmed it with one of our own studies where we analyzed patients with hypercortisolism that sometimes also have a mineralocorticoid-like excess effect, where we also identified the relationship between plasma potassium and phospho-NCC in UEVs. So taking it a step further, this was one of our previous studies where we tried to analyze whether UEV biomarkers could also help to predict a response to therapy. And indeed, using a small number still, this was possible to demonstrate. So this was from a clinical trial where we analyzed the effect of thiazide diuretics in kidney transplant recipients with hypertension. There is evidence that NCC and phospho-NCC can be upregulated, especially by calcineurin inhibitors and tacrolimus. And then it makes sense to inhibit this with thiazide diuretics. But not all recipients responded the same, and we could divide the blood pressure response in responders and non-responders. And then when you analyze at baseline in UEVs phospho-NCC, we were able to show that phospho-NCC was higher in those kidney transplant recipients with hypertension that subsequently responded to thiazide treatment in terms of blood pressure response. So this, we believe, is also interesting and really could turn certain UEV proteins into biomarkers. Still keeping with kidney transplantation, this is another recent study by a German group that I think nicely illustrates also the approach that people now use for these type of studies. Again, using a differential centrifugation to isolate vesicles, using electron microscopy to confirm the presence of vesicles in urine, showing the size distribution and illustrating that indeed this matches with exosomes mostly around 150 nanometers. Also showing with proteomics that all of the nephron segments are present in the proteome. So certain transport proteins of each different tubular segment, but also glomerular proteins and also these exosome-specific proteins. And what they then did is at different time points, they followed the donor urine sample prior to transplantation, and then in the recipient also the urine samples on day 0, 1 month, and 1 year, providing a dynamic range in the UEV proteome in the course of kidney transplantation. And the two main conclusions was that they found a strong dynamic regulation of complement activation, probably due to ischemia reperfusion and acceptance of the allograft, also suggesting that improvements might be made by inhibiting this early complement response. And of interest also is that they identified also in an independent cohort a certain protein, PCK2, as a putative prognostic marker for allograft function, illustrating that indeed it might be possible using UEVs to obtain a non-invasive measure of allograft function that might correlate with future outcomes. Still keeping with complement, we also have an interest in polycystic kidney disease, which is, we believe, also a disease that could benefit from good molecular markers to identify those patients at risk for progression of their disease and deterioration towards kidney failure. This was a cross-sectional analysis in patients with either polycystic kidney disease or patients with chronic kidney disease who were matched by sex, age, and eGFR. And we first employed mass spectrometry, identified a number of targets, and then confirmed these targets using a targeted approach with immunoblotting. And we identified two groups of proteins, pleckins, but also again complement, to be upregulated in patients with polycystic kidney disease. And currently we are taking this a step further using a cohort of patients with stable kidney function and polycystic kidney disease versus those with deteriorating and fast progression of kidney failure to see if these biomarkers might indeed predict future kidney loss in these patients. Then finally, as mentioned, the least evolved but perhaps most intriguing aspect of this field is UEVs as mediators of kidney function and kidney disease. This is a cartoon showing several potential pathways by which cells can take up vesicles. It's still not established whether there's one dominant route or whether there are several possibilities. But at least this cartoon gives an overview of the different possibilities how target cells can take up vesicles and how the cargo of these vesicles might influence cellular function. And this then also allows for the option to, as it is called, engineer exosomes or vesicles with certain payloads to influence and actively influence target cell behavior. So really moving towards therapeutic application of vesicles, which especially in oncology is already emerging. Within nephrology, there are also some signs that there is regulation, for example, hormonal regulation of extracellular vesicle uptake. This is, I thought, a very elegant study where they actually infused systemically extracellular vesicles and then analyzed the recovery in urine of these vesicles, showing that the antidiuretic hormone vasopressin regulates the uptake of extracellular vesicles by the collecting duct. And this was illustrated by interfering with a vasopressin receptor antagonist tolvaptan. And this effect could not be reproduced by using another intervention with a loop diuretic. This is also of interest because this implies that infused extracellular vesicles apparently reach the urine, which also raises the question whether they might be filtered or tubularly secreted, which is, I think, an interesting and unresolved issue. So then the final study I wanted to share is also an earlier study that analyzed the antibacterial properties of urinary extracellular vesicles. And indeed, if you analyze the protein, there are several anti-infection components present in UEVs, also antiviral components to make it a bit more topical. But the elegant aspect of this study, I thought, was that they performed an E. coli lysis assay and found that when E. coli was combined with exosomes for a longer period of time, there was increased lysis of bacteria mediated through the presence of exosomes, indicating that indeed they possess antibacterial properties and that the cargo of these vesicles can affect cells. So in conclusion, I think it is fair to say that urinary extracellular vesicles are a rich source of urinary biomarkers. There are still methodological caveats and limitations, especially being aware of the heterogeneity of EVs and bias introduced by the methodology used. And to resolve this more or less, there is an IMPRESS position paper to which many UEV groups contributed, which is currently in press, and I think will really be helpful to move the field a bit further, also towards clinical application. And finally, I think it has great untapped potential for nephrology in general. And with that, I would like to acknowledge the PhD students in my group who actually did most of the work, the funding by the Dutch Kidney Foundation, and I thank you for listening, and I'm happy to answer your questions. Thank you.
First of all, thank you for your very interesting presentation. So now, let's have questions and comments from the audience. Does anyone have any questions or comments? in the Zoom window at the bottom of the screen, you can find icon menu. So when you speak, please make sure your icon is not muted. Or you can post your question to chat. I look forward to active participation. So on chat, there is one question. So you showed the effect of tolvaptan on urinary vesicles for ADPKD. How about bardoxolone methyl? Have you examined it?
Yes, I saw that question as well. I think it's an excellent question. I think this is the point where the field currently is, that we should apply these methods, especially in clinical trials investigating certain drugs, to see if outcomes of drug effects can be coupled to changes in the UEV proteome. So I don't have the answer yet, but this is, I think, where the field is ready for to apply this method in clinical trials that have urine available to make these analyses and to see whether drug effects on the kidney also translate into differences in UEVs.
So, are there any more questions?
I think some audience are raising their hands, like Dr. Razak, Dr. Kato, and Dr. Muto. You can find it in the list of the audience.
Also, Dr. Muto, please, the question.
And you also have 3 questions.
Where can I find it? Okay, so.
Yes. Shinya, you can also have Q&A. There are 3 questions from audience.
Okay, thank you.
Yeah, I've opened the chat also. I see some questions here. One question is, does the quantity and quality of UEVs change with aging? Which is, I think, a great question, an interesting question. We believe this relationship exists, and we have now one study to analyze whether kidney function and kidney volume or kidney mass or nephron number also determines the number of urinary extracellular vesicles. And we do see this relationship. And therefore, we also think that the quantity of UEVs will change with aging, but that this is probably mostly determined by loss of nephrons, because this is the major source of UEVs.
Thank you very much.
Thank you very much. So also, I think it's time. So also, yeah, let's move on to the next presentation. Dr. Hoorn, thank you.
So thank you very much, Dr. Hoorn, for your excellent presentation. And there are some in the Q&A box who also congratulate for the presentation and for this excellent summary. So we have to move on to the next presenter. So it is again my pleasure to introduce you our next presenter, Dr. Kawari Hayashi from the Keio University, Tokyo. I just would like to say some words about the latest topics of her research, which was like the investigation of metabolic factors associated with EGFR decline, or the epigenetic regulation through the angiotensin blockade in podocytes. And now actually we are going to hear a presentation on the DNA repair, so the DNA damage and the DNA methylation in kidney disease. So please, Dr. Hayashi, keep your presentation. Thank you.
Thank you for giving me an opportunity to talk about my research today. And thank you for your kind introduction. It is a great honor to participate in this meeting today. I'd like to focus my presentation DNA damage repair and DNA methylation in kidney diseases, focusing on podocytes. I have nothing to disclose. And this is the message I'd like to deliver you today. Understanding DNA damage repair and DNA methylation in podocytes may be useful for development of therapeutic strategy against kidney diseases. And I will begin my talk with our previous report about KLF4-mediated DNA methylation in podocyte phenotypic modulation. And then I'd like to talk about DNA damage repair and DNA methylation in podocytes, and then detection of podocyte DNA damage in human samples. Recently, memory effect or legacy effect has been attracting attention. This is a sustained effect of a transient treatment or insult in lifestyle-related diseases, and first demonstrated in large clinical trials of diabetes, such as DCCT or EDIC trials, which has been recognized as metabolic memory. And recently, epigenetic mechanism is suggested to be involved in the metabolic memory. And we have investigated these memory effects, especially the memory stored in the kidney. As you know, epigenetic mechanism is defined as a set of regulatory modifications that influence gene expression above the level of genome itself without changes in gene sequence. As shown in this figure, there are three major mechanisms: DNA methylation, post-translational histone modifications such as histone methylation or acetylation, or RNA-based mechanisms. The epigenetic mechanism is characterized by persistence, as the DNA methylation state is maintained in dividing cells, and also by reversibility, as it can be changed by environmental factors and aging. And recently, epigenome-wide association studied in CKD has been reported that the association of DNA methylation in blood or kidney cells with kidney function. And we are interested in DNA methylation changes in kidney, especially in podocytes. As you know, podocytes is an important cell to form a slit membrane, a barrier for proteinuria. And podocyte damage causes food process effacement, detachment and apoptosis, which causes glomerulosclerosis. And I'd like to talk about our previous report about KLF4-mediated epigenetic changes in podocytes. This slide shows you transient induction of podocyte KLF4 expression by TETON system ameliorates albuminuria with adriamycin nephropathy in mice. In this mice, KLF4 was induced by doxycycline treatment. And interestingly, the albuminuria was decreased not only during the doxycycline treatment, but also after cessation of the treatment. An in vitro study using human cultured podocytes suggested that KLF4 expression causes morphological changes in podocytes and induces epithelial cell markers such as nephrin, podosin, p-cadherin, and WT1, and reduces mesenchymal cell markers, which is induced in podocyte damage such as alpha-SMA and vimentin in RT-PCR analysis. As you know, KLF4 is one of the Yamanaka factors inducing iPS cells and is reported to be involved in epigenetic remodeling. So we investigate the KLF4-mediated epigenetic changes in podocytes using comprehensive DNA methylation array. As a result, KLF4 overexpression induced demethylation of the promoter lesions of the epithelial cell markers such as nephrin, and methylation of the promoter lesions of the mesenchymal cell markers such as vimentin, suggesting that KLF4 is associated with gene-specific DNA methylation changes in podocytes and promoter lesion of podocyte markers. And together with the result of the association between angiotensin II and KLF4, it is suggested that upregulated renin-angiotensin system in CKD induced a decrease in KLF4 expression in podocytes, leading suppression of nephrin expression through increased DNMT1, DNA methyltransferase 1 binding, and DNA methylation of the nephrin promoter lesion. And KLF4 may contribute to the sustained decrease in proteinuria of renin-angiotensin system inhibitors. And next, we consider how do epigenetic changes arise, and focusing on DNA damage repair system, because DNA double-strand breaks may induce DNA hypermethylation in vitro studies, and podocytes are maybe vulnerable to injuries such as DNA damage because of their terminally differentiated and non-dividing features. So we hypothesized that DNA double-strand breaks may induce epigenetic changes leading to disease phenotypes in kidney podocytes. And we focused on DNA repair factor K85 because K85 is an important DNA double-strand break repair factor and is also reported to be an interacting protein of KLF4. To investigate the role of K85 on podocytes, we generated podocyte-specific K85 knockout mice. Interestingly, the knockout mice developed severe focal segmental glomerulosclerosis with large amount of proteinuria, and the knockout mice died around 6 weeks of age because of renal failure. In this mice, DNA double-strand break sites was increased, and also promoted DNA methylation was observed in the knockout mice. And the expression of DNMT1 and DNMT3B was increased in isolated podocytes of the transgenic of the knockout mice. And bisulfite genomic sequencing in the nephrin promoter region suggested that increased DNA methylation was observed in the promoter lesion, which leads decreased expression of the nephrin gene. And chromosomal bonding analysis suggests that long-term K85 knockdown induces DNA double-strand breaks and genome instability, such as chromosomal breaks or double minutes. All these findings suggest that K85 is essential for maintaining in podocyte genome integrity. And to investigate the role of K85 on the decrease in proteinuria, we performed in vitro analysis using human cultured podocytes. K85 overexpression caused increased nephrin expression with decreased DNA methylation and decreased binding of the DNMT1 at the nephrin promoter region. And interestingly, at the same nephrin promoter lesion, decreased DNA double-strand break sites was observed, suggesting that K85-mediated DNA repair may be associated with DNA methylation status. And this is a summary of our findings. We found that K85 is decreased in diabetic nephropathy podocytes. And high glucose causes both the increased DNA damage induction and decreased expression of K85, which causes increased DNA damage sites and increased DNA methylation in the podocyte marker genes such as nephrin, which leads increased proteinuria. And next, we would like to detect and evaluate podocyte DNA double-strand breaks in human samples. We use the method of long-distance PCR method, as previously described, which is based on the assumption that DNA with fewer double-strand break lesions will amplify to a greater extent than more damaged DNA if equal amounts of DNA from different samples are amplified under identical conditions. And this picture shows the immunofluorescent double staining of podocyte marker WT1 and DNA double-strand marker gamma H2AX in a glomerulus with a patient with IgA nephropathy. The number of WT1 and gamma H2AX double positive cells, which means the DNA damaged podocytes, was significantly increased in patients with pathologically diagnosed podocytopathy. On the other hand, long-distance PCR method of nephrin gene using glomerular samples obtained by laser microdissection suggested that increased DNA double-strand breaks in patients with podocytopathy. And together with the fact that DNA damage repair is promoted at opened chromatin and increased chromatin accessibility at the nephrin gene is observed in podocytes because nephrin is a podocyte-specific marker gene. It is suggested that DNA damage in the nephrin gene may reflect podocyte DNA damage. And we next evaluate podocyte DNA double-strand breaks using urine-derived cells. Urine-derived cells contain various types of cells, including podocytes, and long-distance PCR method with long-distance PCR method suggesting that DNA damage in podocytes was increased in patients with hypertension and diabetes compared to those with hypertension alone. And now we are investigating the role of DNA damage on podocytes using IPPO1 transgenic mice. IPPO1 is a homing end nuclease cutting a fixed DNA sequence. In this transgenic mice showed increased albuminuria with podocyte morphological changes such as food process effacement. And to investigate the association of DNA damage repair and DNA methylation, we performed MEDIPSEC analysis using isolated podocyte in the transgenic mice and wild-type mice. The methylation comparisons of DMR's differentially methylated regions suggested that increased DNA methylation in IPPO1 mice, this blue line, was observed in CPG islands, gene bodies, and promoter lesions. And interestingly, the methylation changes was not significantly correlated with the distance from IPPO1 cutting site, suggesting that there may be a mechanism of DNA methylation independent of the DNA damage site. So we are now investigating the molecular mechanism. This is the final picture of my talk. This picture shows the spectrum of podocyte responses after injury. Depending on the severity and duration of the injury, podocyte may respond to the injurious stimuli such as hypertrophy, EMT, and their differentiation, or detachment anaptosis, which is associated with increased DNA damage accumulation and altered DNA methylation. And these may be potential prognostic markers or hopeful therapeutic targets for chronic kidney disease through epigenetic phenotypic modulation of podocytes. And we consider focusing DNA damage repair factors has an advantage because DNA damage repair system is cell type specific. So this may be a potential therapeutic target through epigenetic modulation with fewer side effects. In summary, understanding DNA damage repair and DNA methylation in podocytes may be useful for development of therapeutic strategy against kidney diseases. I'd like to acknowledge all the collaborators and all the members of the laboratory. Thank you for your attention.
Thank you very much, Dr. Hayashi, for your excellent presentation and for your excellent summary, and also thank you for giving the time, because now we have 5 minutes left to the questions, and we have one question in the chat box and one in the Q&A box. So the first one is in the Q&A box is coming from Tomofumi Moriyama, who is asking about that. In the future, can we evaluate podocytes function and select treatment strategy by genetic analysis? That's the question. What do you think about that?
Yes, thank you for your important question. I consider in the future podocyte analysis using the urine-derived cells is very important, both epigenetic and genetic evaluation.
Thank you.
Thank you.
Thank you. And we have another question in the chat box also for you. So if I understand it well, so the one who is asking this question is inquiring about the possibilities in the field of drug discovery. So she's just asking that when we think about a drug discovery, we have to do a systemic administration. So do you have some idea about that? So what can be the role of epigenetics and DNA methylation related to this field? Thank you.
Thank you for an important question. There are some epigenetic modification drugs already available in clinical use, such as hematological malignancies, and considering the application of these drugs for CKD treatment, the problem of the side effect is maybe very important. So as I mentioned in the presentation, focusing on the DNA damage repair system has an advantage because the DNA damage repair system is cell type specific, so it is less side effects. And alternatively, there may be a way to use the low dose of epigenetic modification drugs in combination with the for example, renin-angiotensin inhibitors for expecting the additive effects. Thank you.
Thank you. That's very important. So hopefully it can be a kind of biomarker also for the early diagnosis of non-communicable diseases like DND, so diabetic nephropathy, and so on. So that's how it can be also a kind of part of the drug discovery. So may I just ask from the panelists if there is anyone who would like to join to the discussion or who have any question related to this presentation. And I also checking the Q&A box also. So we have one congratulation from your colleague. There is no question. There's another one. How do you use normalization when measuring DNA DSB? That's the question.
Thank you for your important question. As you mentioned, the amount of total DNA is important to evaluate the evaluate DNA double-strand breaks using the long-distance PCR method. So in the result of our study, the kidney function are similar between these groups around EGFR 60, and the expression of podocyte cell markers such as nephrin and podosin were similar between the groups. So, but the evaluation, the number and the amount of podocytes in the samples is very important. So we're now investigating how to evaluate and count the podocyte containing in the urine samples. Thank you.
Thank you very much. And finally, if there's no other questions, I cannot see any raised hands or any messages in the Q&A box. So I would like to honestly congratulate you for your research and for your presentation. And I would like to ask my co-chair, Dr. Nakatani, to present the next presenter. So thank you very much, Dr.
Thank you very much.
Thank you, Dr. Hayashi. Thank you for a nice talk. Next, the first speaker of the clinical session is Professor Jane from Cambridge University. As you know, his specialty is the ANCA-associated vasculitis. The title of presentation today is Similarities and Differences of ANCA-associated vasculitis in Japan and Europe. So, Professor Jane, thank you. Could you start the presentation?
Thank you very much. And can you hear me and see my slides?
So we can.
Good.
Yes, thank you.
So I'm very grateful to the Japanese Society of Nephrology and European Renal Association for inviting me to give this talk. And it's been an interesting reflection on almost 20 years of collaboration with many Japanese colleagues in nephrology and rheumatology. So I'm based in Cambridge in the United Kingdom, which is about an hour and a half north of London. And this is a photograph of our enlarging campus in Cambridge. In my talk, I'm going to touch on nomenclature and classification, clinical epidemiology and phenotype, and then treatment and outcome, with the perspective of Japanese-European interaction and collaboration. This is just some timelines of the collaborations, the formal collaborations that have been undertaken. And it really started in the early 2000s with a grant from Japan Health Sciences Foundation to study the epidemiology of vasculitis in Japan. And a group of colleagues from Europe who had undertaken similar work before were invited to join this. As a result of that, we went on to discuss and compare phenotypes and exchange research fellows. And then we started to do some joint work on interventions such as IVIG and rituximab. And that led to collaborations on international randomized controlled trials, the plasma exchange, rituximab and avacopan most recently. And I think it's important to note the contribution that Japanese physicians have made towards major meetings, the Asia Pacific Vasculitis meeting in 2012 and the 18th International Vasculitis Workshop in Tokyo in 2017. And Japan has become very much a leader in vasculitis research. But the background to this is one of quite rapid change in nomenclature and classification. And we were aware in the 1980s and early 1990s of major differences between Europe and North America, which led to consensus statements in the 1990s. And then in the early 2000s, considerable differences between Japan and North America and Europe in the Japanese approach to classification, for example, of renal vasculitis and microscopic polyangiitis. And this ultimately led to Japanese involvement in the Chapel Hill Consensus Statement of 2012, which is the current state of the art for the nomenclature and the first time that the ANCA-associated vasculitis subgroup was clearly defined. So this represented something of a landmark in international collaboration. We were aware that there were some differences in the epidemiology and phenotype between of renal vasculitis between Japan and the UK. And this was a small initial study conducted in the early 2000s in which there were no cases of Wegener's, now GPA, or Churg-Strauss syndrome reported by nephrologists in the centers in Japan that we were talking to. And the dominant diagnosis was microscopic polyangiitis. If we looked at serology, we found, again, the dominant ANCA serotype was myeloperoxidase, over 90% of patients, with no patients in Japan with renal vasculitis being diagnosed with PR3 ANCA positivity. Whereas in Europe, for renal vasculitis, we see around 55-33% split. It transpired that the reason for this was that the proteinase 3 ANCA assay was not reimbursed at the time by the Japanese healthcare systems, whereas myeloperoxidase assays were. So it simply was not being looked for. This then inspired a study of the performance of ANCA assays in Japan and Europe. And essentially, we found that the ANCA assays performed extremely well in Japan, both for MPO and PR3, with high rates of sensitivity and specificity for MPA and GPA. And of the 3 MPO ANCA commercial assays being used in Japan at the time, there was really a very good correlation between them. So these differences in ANCA frequency were not a feature of assay performance, which was good. We then went on with an epidemiology study led by Hiroshi Hashimoto from Juntendo and Shuichi Fujimoto from Miyazaki. And now, with the inclusion of the PR3 ANCA assay, we were detecting patients who are PR3 positive, but still a much lower frequency, 7%, compared in this study to around 58% in a European cohort. Also, the dominant diagnosis was microscopic polyangiitis in Japan, with an incidence of about 18 per million per year, much less frequently in Europe. But if you were to combine GPA, Churg-Strauss, now eGPA, together with MPA, the total incidence of ANCA vasculitis was pretty well the same, at just over 20 per million per year. This is looking at all presentations, renal and non-renal presentations. When we looked at organ involvement, I'm going to say a little bit more about MPA in a moment, but it was striking that GPA in Japan, albeit rare, had less frequent involvement of the kidney and less frequent involvement of the lung, and was very much driven by the ear, nose, and throat system in terms of diagnosis and manifestation. So this led on to a more detailed comparison of phenotype and outcome of microscopic polyangiitis, as I've mentioned, the dominant phenotype in Japan. And we compared several cohorts. So this was a cohort from Cambridge compared to quite a large cohort from Japan and a UVAS cohort, which is historic clinical trial patients. And I'm just highlighting some of the differences. Age at onset of MPA in Europe is fairly consistently around 65 years of age. And in Japan, it's around 5 years older. As I've already mentioned, myeloperoxidase is the dominant serotype in Japan, less so in Europe. Serum creatinine tends to be higher in Europe. The UVAS studies were biased because we were selecting patients with more severe disease, but nevertheless, between these two cohorts, Japanese patients tended to have less severe renal dysfunction at diagnosis. When we looked at the patterns of renal involvement, and this slide shows different cohorts looking at different organ manifestations, we saw more skin and eye disease in Europe and more frequent neurological and chest disease in Japan. And it's particularly, the black line is standing for interstitial pneumonitis, is really quite common in patients presenting with MPA in Japan, and I'll say a little bit more about that in a moment. When we looked at the outcome of these patients, so this is looking at renal survival over 5 years, there was really no major differences. So the Japanese patients, as I said, tend to be slightly older, but tend to have slightly less bad renal function, but this sort of balances out in terms of long-term outcome. We did, however, look at treatment, and it's important to appreciate that treatment practice is changing in Europe and treatment practice was changing in Japan. But this was a sort of snapshot around 10 years ago when cyclophosphamide use was less common in Japan than Europe. Steroids were pretty well universal. Azathioprine use was also much less common in Japan compared to Europe. Mesorabine is a drug that we don't have access to in Europe and is used by some patients in Japan. And at the time we did this study, rituximab was not yet available, or at least not being widely used in Japan. So this was a snapshot at the time. When we did a multivariate analysis looking across the pooling of these cohorts for risk factors for end-stage renal disease, no surprises that baseline serum creatinine is the dominant risk factor. But we also found that immunosuppressive use, particularly with cyclophosphamide, reduced risk of end-stage renal failure. In this study, plasma exchange didn't seem to help, neither did an intravenous steroid. It's hard to put too much on this data, except to highlight that there were quite significant differences in practice at the time. We went on to look at a GPA, formerly Wegener's, using a similar type of analysis. And whereas in UK, fewer than 10% of GPA patients are MPO ANCA positive, in Japan, 34% of GPA patients were ANCA positive. Also, as I alluded to earlier, renal disease is less common, reflected in a lower serum creatinine in the Japanese cohort compared to the UK cohort. Just taking an aside to the lung, and I don't apologize for talking about this to a renal audience because we're talking about a multi-system disease. We worked with Japanese colleagues on a systematic study of CT appearances in the lung in patients with microscopic polyangiitis. And the take home from this study is that the radiological appearances of disease are common, with 2/3 of Japanese patients having evidence of interstitial lung disease, similar frequency with airway lesions, and several other types of pathology were seen. We've done similar studies in the UK, and the frequency of these lesions is around 1/3 of these rates. So we see around 20% with interstitial lung disease, 20% with airway lesions, and 20% with other types of pathology. So there are major differences between Japan and UK in terms of the pulmonary disease. And this is not just true in vasculitis, it's also reported in other inflammatory syndromes. I think the take home again for junior colleagues on the call is the importance of CT scanning all patients with a renal vasculitis with a number of different appearances that can be seen.
Biopsy carries a prognostic value.
Oops.
And the simplest...
Sorry about this. This is a pre-recorded slide. Here I'm talking about the histological classification of vasculitis. And in Europe, we've developed the so-called Bearden scheme, defined by 4 different categories, looking at glomerular pathology. A similar study has been done in Japan, where they've looked at the same Bearden classifications of focal, mixed, crescentic, and sclerotic. Overall, this is quite a small study, but the overall outcomes were much better than we see in Europe, although follow-up in this study was shorter, but with those patients with a so-called focal or mixed pathology really doing very well. Now, in this study, they reviewed all of the different cohorts of renal biopsies looking at prognosis in ANCA-associated vasculitis. And there are some trends. And one of the trends, I've highlighted here the Japanese cohorts that have been reported. One of the trends is probably a higher proportion of patients with focal disease, which is the most benign lesion, fewer than 50% of glomeruli affected in the Japanese cohorts. This either means that the Japanese physicians are good at picking this disease up very early, or it means the disease is pursuing a slightly more benign course. Moving on to therapy, we started studying rituximab in ANCA-associated vasculitis in the early 2000s. And we assisted with the first use of rituximab in Tokyo, which is in 2006, in patients with refractory Wegener's. As a result of this interaction and working together subsequently, Japanese colleagues have made a major contribution to a recent study called Retazarem, looking at rituximab remission therapy in ANCA vasculitis, for which we've reported the results at a recent ERA last year. And Japanese investigators are taking this further to do their own study, trying to minimize glucocorticoid exposure by using rituximab as induction for ANCA vasculitis in a study called LOVAS, which is shortly to be reported in detail. So in summary, through the 2000s, there was really a harmonization of nomenclature and classification, and this is critical if you're going to start doing combined clinical studies or clinical trials. The overall incidence of ANCA vasculitis is similar between Europe and Japan, but MPO-ANCA is the dominant serotype, 85% in Japan, and microscopic polyangiitis the dominant diagnostic subgroup. Japanese patients tend to be older, have less severe renal involvement, and more lung disease than European patients, and I've pointed out the importance of pulmonary CT. I think it's hard to draw firm conclusions about management because practices are changing, but there were definitely differences in use of immunosuppressives, although overall outcomes appeared similar. Uptake of rituximab is now changing practice considerably. And I think the sort of proof of success of this long-term interaction has been the collaboration in designing and conducting randomized controlled trials in ANCA vasculitis. And this is a slide just showing the Pexivas steering group. This was a study of plasma exchange, who met in Kyoto in 2012 while launching the study. I'd particularly like to acknowledge Kazuo Suzuki, who really pioneered the interaction between Japanese vasculitis colleagues and Europe, and Toshiko Ihara, who has really worked very hard, particularly on the randomized trials in Japan, and many other colleagues in Japan and the European colleagues in the UVAS group. Thank you for your attention.
Professor Jane, thank you very much for the informative presentation. So first I have a question, and I understand that rituximab is very useful. So on the other hand, the immunosuppressive side effect is a serious concern. In this COVID-19 pandemic situation, could you let me know how to use rituximab for patients with ANCA-associated vasculitis? should we use it after vaccination? And the question on chat is post-COVID recovery, we can give rituximab after how long, please?
Thank you. Very topical question, and one that is really complicating our management of ANCA vasculitis, because rituximab is our preferred induction and maintenance agent now for ANCA vasculitis. If you have time, patients should definitely receive both doses of a vaccine prior to rituximab. And the problem is if you don't have time, and if you don't have time, your choices are either to use an alternative drug, such as cyclophosphamide, or maybe steroids alone if it's a mild disease, or to give the rituximab and vaccinate anyway. Our data from Cambridge, not yet published, is that patients with who have had rituximab have an antibody response to COVID vaccine that is less than 50% that you would see in healthy controls, and some are not responding at all. So I think the concern about rituximab and vaccination is absolutely true, but you have to balance the risks of controlling the vasculitis and exposing patients, say, to prolonged high-dose steroids, as opposed to the risks of a failure of COVID immunization. And I don't think there's an easy answer. And I think for each patient, you have to make what you think is going to be the correct judgment.
Okay, thank you. The next question is Dr. Guchi. Thank you. Do you think the difference in ANCA vasculitis between Japan and Europe due to genomic differences or environmental differences? Genomic or environmental?
I think it's genomic, but I don't think we've had the opportunity to do the studies in sufficient detail to confirm this. The reason I say that is that we have good data that the immunogenomics, particularly HLA class 2 associations, are closely associated with the autoantibody. And because the MPO autoantibody is dominant in Japan, that's likely to be driven by underlying immunogenetic susceptibility. But we would like to do comparative genomic studies, but they have not been conducted yet.
Okay, so thank you. The last question is, thank you for excellently showing that there are significant differences between Europe and Japanese ANCA vasculitis patients. What is the pathological reason for these differences? This question is about pathological issue.
Yeah, so there are subtle differences in the renal histology between Japan and Europe, and these have not been formally compared in a prospective study, but we've had many meetings with renal pathologists in Japan. I mentioned that the focal phenotype is slightly more common in Japan, but another feature that we see more frequently in Japan is actually extraglomerular arteritis, which is really quite rare in European biopsies, but is more common in Japan and is completely unexplained why that should be more frequent. Those are the sort of two differences that come to mind. But again, we have a lack of good quality comparative data.
Okay. Professor James, thank you for your nice talk. I'm afraid there's no time for any questions. So I'd like to move on the next presentation.
Thank you very much. And Professor Jane, thank you very much for your excellent presentation and review. Now we are moving on for the next presenter. So let me just introduce you, Dr. Yusuke Sagaguchi from the University of Osaka. And let me just say some words about their previous researches. So our colleague just studied the erythropoietin stimulating agents and mortality among hemodialysis patients, and later on the possible role of hypophosphatemia in hemodialysis patient cohort. And now we are going to hear his presentation on the emerging role of hypomagnesemia in chronic kidney disease patients. So please, Dr. Sagaguchi, keep your presentation. Thank you.
Thank you. Thank you very much for your introduction. My name is Yusuke Sagaguchi from Osaka University. I really appreciate all moderators of JSN and YNP inviting me to this exciting meeting. And my topic today will be about the role of mechanism in CKD. These are my disclosures relevant to my presentation today. First of all, we should be aware that hypomagnesemia is not rare among patients with CKD. We have previously reported that hypomagnesemia, as indicated by red bars in this figure, is one of the most frequent electrolyte abnormalities among CKD patients not on dialysis. Notably, its prevalence was not reduced in the advanced CKD stage 4 and 5, where its prevalence was about 15%. There are many causes why CKD patients develop hypomagnesemia, such as diabetes, massive proteinuria, frequent use of diuretics, and maybe tubular dysfunction, which leads to urinary magnesium wasting. But what is the clinical implication of hypomagnesemia in CKD patients? About 10 years ago, I got curious about magnesium in CKD patients and examined an association between serum magnesium levels and the risk of end-stage kidney disease. In this retrospective cohort study of patients with type 2 diabetes, lower serum magnesium levels, defined as less than or equal to 1.8 milligram per dl, was strongly associated with higher risk of initiation of renal replacement therapy. Therefore, low magnesium might be involved in the progression of kidney diseases. Another more important aspect of magnesium is its impact on vascular calcification. In this in vitro study coming from Netherlands, you can see in the middle panel that high phosphate, in this case 10 millimolar phosphate, induces calcification of vascular smooth muscle cells in a time-dependent manner as expected. What is surprising here is shown in the right panel that this calcification was prevented almost entirely when increasing magnesium concentration to 2 millimolar. This anti-calcification property of magnesium has been confirmed repeatedly by several other groups. And in line with these in vitro studies, in vivo experiments also found the similar protective effect of magnesium on vascular calcification. Spanish researchers have found that in nephrectomized rats, high phosphate diet induces severe aortic calcification, as we well know, but this calcification was largely attenuated by high magnesium diet, as shown here. A similar finding was observed in colossal knockout mice. We can see that aortic calcification was again remarkably prevented by high magnesium diet. Therefore, these studies have strongly suggested the benefit of magnesium in the prevention of vascular calcification. Here, we conducted a randomized control trial to examine whether magnesium really inhibits the progression of vascular calcification in pre-dialysis CKD patients. This is a 2-year open-label randomized control trial including stage 3 to 4 CKD patients with some of the risk factors for coronary artery calcification, that is, diabetes, prior history of cardiovascular diseases, high LDL cholesterol, or current smoking. All patients underwent coronary CT at baseline and 2 years later. And the primary outcome of this trial was the % change in the Gaston score, which indicates the amount of calcification and the coronary arteries detected by coronary CT. The doses of oral magnesium oxide were adjusted every 1 to 3 months to achieve serum magnesium levels at least 2.5 mg per dl. We randomized a total of 123 CKD patients to the magnesium oxide group or the control group in a 1-to-1 ratio. During the 2-year follow-up period, several patients dropped out from the study, including 3 patients with diarrhea in the magnesium oxide group. At the end of the study, there were 46 patients in the magnesium oxide group and 50 patients in the control group, and these patients were finally included in the primary outcome analysis. The baseline characteristics of the 2 study groups are shown here. The mean age was about 70. 3/4 of patients had diabetes, and half of the patients had prior cardiovascular diseases. The mean eGFR was about 35, and the mean magnesium level was 2.0 in the magnesium oxide group and 2.1 in the control group. The median Gaston score was about 200, indicating that many of these patients have moderate coronary calcification. As a whole, the baseline characteristics were well balanced between the 2 groups. Here is the result of the primary outcome. There was a significant difference in the % change in the Gaston score between the 2 study groups. The % change in the Gaston score was about 11% in the magnesium oxide group, which was significantly lower when compared to the control group, which showed a 40% increase. The between group difference was irrespective of the baseline Gaston scores, less than 400 or greater than 400. This is the first study to show the clinical efficacy of magnesium on coronary artery calcification in CKD patients. This slide shows the change in serum magnesium levels during the study period. Serum magnesium levels were significantly elevated in the magnesium oxide group from 2.0 at baseline to 2.3 at the end of the study. Even though this value was slightly lower than the pre-planned target level of 2.5, the significant effect of magnesium was still observed in this trial. The average dose of magnesium oxide in this trial was about 500 mg per day. So what is the underlying mechanism of the anti-calcification property of magnesium? Here, calcium protein particles, CPPs, play a pivotal role. As illustrated in this figure, calcium and phosphate in the circulation bind with phytoin A, which forms soluble calcium protein particles, CPPs. CPPs usually exist as CPP1, which contain amorphous calcium phosphate. However, when serum phosphate levels are elevated in CKD patients, CPPs spontaneously undergo topological changes from CPP1 to CPP2, in which calcium and phosphate are highly crystallized. Notably, among these two types of CPPs, CPP2 are suggested to induce vascular calcification. This very important evidence from Switzerland has shown that CPP1 have little effect for vascular smooth muscle cells, whereas CPP2 strongly induce calcification of these cells. Therefore, CPP2 are considered to be the major inducer of calcification, although in vivo effect of CPP2 is not yet clarified. Interestingly, magnesium is known to strongly inhibit the crystallization of calcium and phosphate and impair the maturation of CPP1 to CPP2, at least in vitro experiments. As shown here, 2 millimolar magnesium completely inhibits the in vitro crystallization of calcium and phosphate. Thus, it is considered that the effect of magnesium on CPPs, as shown here, explains at least partly the anti-calcification property of magnesium. And this hypothesis was tested by a recent randomized trial of hemodialysis patients. This trial examined how dialysate magnesium concentrations affect serum calcification propensity, known as T50. T50 was developed by Dr. Pash, as displayed here. I don't want to go into detail about T50, but T50 actually measures inhibitory capacity of patient's serum against the maturation of CPP. And in this particular trial, T50 was significantly elevated when patients were dialysed with a high magnesium dialysate, that is 2 milliequivalent per liter. This means that high magnesium dialysate restores the inhibitory capacity of patient's serum against the maturation of CPP. This is inconsistent with the in vitro findings, as I have shown in the previous slide. What is more interesting in this trial is that the researchers with this study further examined inflammatory cytokine levels, and they found that high magnesium dialysate significantly decrease cytokine levels such as TNF-alpha as well as IL-6. This anti-inflammatory effect of magnesium may also be through the inhibition of the CPP maturation, given that CPP2 is known to induce inflammation when exposed to macrophages, as well as endothelial cells. Based on these findings, we hypothesized that magnesium may be beneficial with respect to hard outcomes. Here, we analyzed Japanese registry of dialysis patients provided by the Japanese Society for Dialysis Therapy, which includes nearly all hemodialysis patients in Japan. And we found that lower serum magnesium levels are independently associated with a higher risk of mortality. According to this observational study, patients with serum magnesium levels between 2.7 to 3.0 show the best survival. And when we look at the combined effect of magnesium and phosphate, there's a significant interaction between magnesium and phosphate. You can see a clear association between phosphate levels and the risk of cardiovascular mortality among patients with lower magnesium levels subgroup. However, this association was attenuated in patients with higher serum magnesium levels and almost disappeared in patients with high magnesium levels. This observational finding suggests that magnesium may be useful, especially for patients with hyperphosphatemia. Finally, we must pay attention to the risk of fracture because excess magnesium may be harmful to the bone, considering that magnesium might impair bone mineralization. But as shown here, we observed a negative association between serum magnesium levels and the risk of hip fracture among Japanese hemodialysis patients, at least when serum magnesium levels were less than 4 mg per dl. Thus, there appears to be less concern with this level of slight hypermagnesemia in terms of the risk of fracture, although a longer-term effect of magnesium should be further studied. Having said that, in clinical practice, we should be cautious about severe hypermagnesemia, such as greater than 5 mg per dl. In summary, a randomized trial has shown the efficacy of magnesium on coronary artery calcification in CKD patients. There's an association between lower serum magnesium levels and a higher risk of mortality among hemodialysis patients. Future randomized trials are required to clarify the long-term benefit of magnesium on hard outcomes. And finally, let me tell you a little bit about Japanese kanji. Magnesium can be expressed in Japanese kanji like this. The left part represents metal, and the right part means beautiful and glorious. I hope this beautiful mineral will pave the way to improve the prognosis of CKD patients. Thank you for your attention.
Thank you very much, Dr. Sagaguchi. It was very, very impressive, and I think this picture can give us a kind of other point of view to observe the magnesium and the role of magnesium as a kind of biomarker, you know, in the cardiovascular mortality and morbidity, I think, because for the last, as you mentioned also, in the last 10, 15 years, we just dealt a lot with hypomagnesemia, and it is quite, surprising that by far, or thus far, for this day, we cannot use the magnesium as a kind of therapy before dialysis or during dialysis. So what do you think about that? if I may ask the first question before the member of the panelists. So what do you think about that? So it is the present in Japan. It can it be a future to administer the magnesium as a kind of preventive therapy?
I think first of all, researchers studying magnesium is very little. So it is very difficult to apply this very important mineral into the real clinical practice. However, we have some magnesium agents such as magnesium oxide for laxative. And also in European countries, you have magnesium containing phosphate binders, probably magnesium chloride. And so we have many opportunities in the current clinical practice to provide magnesium with CKD patients. So we should have more interest in magnesium and should make more evidence about that. Thank you very much.
Thank you very much. I'm asking for the panelists if there is anyone who would like to join to the discussion. So while you are just thinking about the questions, I have found some in the Q&A box. So it is one remark mainly that, so there is someone who just remark or note that, but there is no magnesium in the dialysis fluids in the everyday practice. So what do you think about that? Can it be a practice in this as a standard of care? And the other part of the question relates to the administration of proton pump inhibitors, which also can cause hypomagnesemia and also can worsen the level of hypomagnesemia, for example. So what do you think?
Thank you very much. Both of these questions are very important. And first of all, the first question that the dialysate magnesium concentration is actually very low compared to the ionized magnesium levels of hemodialysis patients. And this means that during the dialysis session, hemodialysis patients experience very low ionized magnesium levels, which may cause of arrhythmic events or muscle cramps, for example. So we need to use or reconsider the optimal magnesium concentration in dialysis. This is a very important question, I think. And the latter question about PPIs, it is very important side effect of PPI, there is hypomagnesemia. And the mechanism of hypomagnesemia induced by PPI is malabsorption of the magnesium in the intestinal or colon. So this can be happen even in the hemodialysis patients. PPI can be a cause of hypomagnesemia, so we should be careful when we have some patients with hypomagnesemia and using PPI. It may be important to stop PPI and change, for example, its blocker and see the reaction about the magnesium level. Thank you very much.
Thank you. It's very, very excellent. Thank you very much. I'm asking the panelists if there is anyone who would like to ask or join to the discussion. We have also 2 or 3 questions in the Q&A box left. Okay, so then let's just move on to the next question. So Dr. Miho Murashima is asking whether, do you think there is any difference in providing magnesium through higher dialysate magnesium concentration versus oral magnesium? So we just touched this question a little bit earlier, but could you please just add something on this issue? Thank you.
Thank you very much for the very important question. I agree with this question. So we need to avoid the sudden drop of magnesium, like potassium, during dialysis session. And while oral magnesium supplementation may be useful for many of the patients, there are several patients who are intolerant to magnesium oxide because of diarrhea. And for such patients, increasing dialysis to magnesium concentration is still a very important option. Yeah.
Thank you.
Thank you.
Thank you. And they have, yes, 2 or 3 more questions more, but 2 or 3 minutes more, not more than 2 minutes, I guess. So let me just read you the last question from the Q&A box. There are also a lot of congratulations for you. And so the last question is that in your intervention study, baseline coronary artery calcification score was higher in the magnesium oxide treated group. Do you think regression toward the mean may have played a role? So can it be a kind of bias? That's the question.
Yes, that's right. But between group difference between magnesium oxide group and the control group is, I think, big enough to return such a normalization to the mean bias, maybe.
Yes. Yeah.
Okay.
So the possible relation between hypomagnesemia and CAC score is highly defendable according to those data, yes. So I'm just asking if there is anyone from the panelists. If there is no questions, I would like to thank you for the presenters, for the excellent presentations, and I give I give the words for Dr. Nakatani to close this presentation session. Thank you very much.
Thank you very much.
Thank you. So last, I'd like to introduce Professor Nangaku from Tokyo University. So Professor Nangaku, would you be kind enough to make a closing remark, please?
Thank you, Dr. Nakatani, to introduce me. We are really impressed with this seminar planned by the Young Nephrologist platform of ERA-EDTA and JSN. I would like to thank the audience, all the chairs and speakers, and especially Professor Martin de Borst and Professor Motoko Yanagita for their devotion. We had more than 200 attendees, and of course, this successful seminar had not been possible without a strong leadership by the presidents of both societies, Professor Christoph Wanner and Professor Naoki Kashihara. Young nephrologists are the future of nephrology, and we are lucky to have great presidents who have strong and enthusiastic desire to encourage young nephrologists. They are very happy to support young nephrologists. I wish this wonderful collaboration between ERA-EDTA and JSN will persist for a long time, and I believe it will. Thank you for joining us today. Please stay safe and healthy. Now let's close this excellent symposium. Thank you.
Thank you very much, Professor Nangaku. I guess that will be all for today. Thank you for taking part. I'd also like to thank everyone who helped us to organize this seminar. Thank you.
Thank you very much, Prof.
Thank you. Bye.
Bye-bye.
Thank you. Bye-bye.